Hello and welcome to Biomentors. Today we are going to discuss about basics of developmental biology, and this would be our second video discussing about the developmental processes and the general terms. So today we are going to talk about potency and stem cell biology. So let's get started. So first of all, what is potency? The potency of cell is an intrinsic property, which means it is an internal property and greater than or equal to its fate. So potency is always equal to or greater than its fate. Cell potency is a cell ability to differentiate, or we can say it is a ability to differentiate into different types of cells or other types of cells. So there are different types of potency. We will see there are majorly 4 types of potency: pluripotency, totipotency, multipotency, and unipotency. So pluripotency means the cell can divide it into any cell type of the body. So if that cell wants to be a muscle cell, it can be muscle cell. If that cell wants to be a blood cells, then it will go in the formation of blood cells. So it can form any cell of the body. All cells, embryonic as well as extra embryonic cells, can be formed by totipotent cells. They can form entire body also, entire bodily cells. What is pluripotency? Pluripotent cells or pluripotency can form. Pluripotent cells can only form the cells of embryo. Means extra embryonic cells cannot be formed. So during the embryo formation, all cells are pluripotent. Pluri means many. Okay. So means, for example, embryo cells will form 3 germ layers, and those 3 germ layer cells will form the entire body or entire development of from that only the entire development of an organism occurs. So all the cells of embryo are pluripotent, and they are potent to form the 3 germ layers: ectoderm, endoderm, and mesoderm. So we can tell that pluripotent cell can form many things. Not everything, not anything, but many things can be formed from pluripotent cells. Now, what is multipotency? Multipotent cells can form closely related cells. So then neighboring cells can be formed. For example, hematopoietic stem cells are multipotent cells because they can form all type of blood cells. Whether it is RBC, WBC, platelets, all type of blood cells are formed by hematopoietic stem cells, right? So they are called as multipotent cells. Few things are formed: neighboring cells or cells which are going to perform a particular function. Structurally they can be similar, but majorly functionally similar cells can be considered as multipotent cells. What are unipotent cells? Unipotent cells are those cells that can only form one type of cells. So one thing is there. Okay, so we will see that totipotency, pluripotency, and multipotency. As we go from left to right, DNA methylation in the cell increases consistently. Okay. So now these are the 4 types of potency, and this was all about potency of a cell. Now we will discuss about stem cells. So what are stem cells? The stem cells have 2 properties. The first property is that they can divide. They have the property of division and self-renewal property. So they have division property, they are actively dividing cells, and they can make self-renewal. So they are stem cells, and The stem cells, the type of stem cells formed by the division can be when the stem cell divides, they form two type of cells, either committed cells, or always they will form a stem cell will divide and then it will form two cells. The first cell is committed cells, so they will go into the commitment, and the second cells which are formed are self renewal cells, same as the stem cells. Then further, this self renewal cells or the stem cell will divide into two cells. One would be committed and second would be self renewal cells, and this cycle continues. So regulative development, a stem cell can divide and in doing so produces a replica of itself, which I have already mentioned. The process is called as self-replication, as well as daughter cell, and the second daughter cell would be committed, as well as daughter cell that can undergo further development and differentiation. So two cells, so suppose this is a stem cell, alright, so it will form two cells. One would be committed cells and the second one will be self-replicating cells. Alright, so this would be self-replicating cells. Then again this cell will divide and will form committed cells, and the second one will again form self-replicating cells. Again it will form. Alright, so this is the general picture how the stem cell will form, always undergo two types of division. Right, the stem cells, and remember stem cells are always undifferentiated cells. Even though their fate is decided, they are undifferentiated cells. Stem cell division. So there are, on the basis of the division, the stem cells can be divided into two types. The first is symmetrical division, symmetrical division, and the second is asymmetrical division. So what is symmetrical division? In symmetrical division produces two self-renewing cells, the stem cells and the daughter cells. So the definition and the which is normal. So either it will form two stem cells or either it will form two committed cells. Either of one would be formed, but it will not form one stem cell and one committed cell. When this condition occurs, it is called as symmetrical division of stem cells. Okay, and this type of thing is rarely seen, and it occurs when if body doesn't need stem cells, because there are already a large number of stem cells in the body, then it will not form stem cells, either it will form committed cells. And for example, body needs more stem cells instead of committed cells, then it will be start producing stem cells, and this occurs very rarely. In some cases it occurs, but the asymmetrical division is a type of division in which normal cell formation is same, which means one stem cell is produced and second is committed cells. Mostly seen, this only mostly occur. The stem cell will divide and form stem cell and a committed cells. Alright, so this was all about stem cells. Now we will see the relationship between potency and the stem cells. So during the formation, when does the when in different cell stages, different type of potency are seen. So we will see from zygotic cells are always totipotent cells. So 4 to 8 cells of zygote. When the first cleavage occurs, all the cells are totipotent cells. After this division, after the division of the zygotic cells, tropoblast and inner cell mass is formed. The tropoblast is an outer covering, and they generally do not participate in formation of in the development of animal. But the inner mass of cells are pluripotent, which means they are not totipotent. They cannot form all type of cells because their fate is determined. Okay, and cell goes from totipotent to pluripotent. From determined, it will either and then after determination occurs, and from the determination stage, after the pluripotent stage, determination occurs, and then after it will either be multipotent cell or it can either be unipotent cells. All right, this was all about potency and potency at the different stages of zygote. Now stem cells, type of stem cells. So these were the two types of stem cells on the basis of cell division, but there are different type of stem cells also. So they are embryonic stem cells and adult stem cells. What are embryonic stem cells? True stem cells can form any cells of embryo. So if we have seen that if the cell can form any cell of embryo, then such type of cell is called as pluripotent cells. So embryonic stem cells are always pluripotent in nature, while the adult stem cells, tissue specific stem cells, they are tissue specific stem cells, tissue repair due to injury or disease. So they are multipotent or unipotent stem cells. They are never pluripotent in nature. So always remember these two terms. Embryonic stem cells are pluripotent and adult stem cells are So this was all about the types of stem cells. Now note, there is a note where in adult stem cells are there and it goes into commitment, and then it would be a transition state where, for example, there would be a transition state for lymphoid progenitor cells or lymphoid progenitor cells, and then myelinoid progenitor cells, and then it will form into lymph tissues, and then it will form into myelinoid tissues. So there would be always a progenitor cell after the commitment of the adult stem cells. They are not capable of self-renewal. Remember, progenitor cells are not capable of self-renewal. Now, induced pluripotent cells. What are induced pluripotent cells? So, according to research by Yamanaka, a scientist from, I think, Japan or China. So, the induced pluripotent cells are cells which are which are unipotent or multipotent, but we want to form it revert back, and we want to form it into pluripotent cells. Such stem cells are called as induced pluripotent stem cells, and they are also termed as Yamanaka factors. And generally these pluripotent cells, the thing which is used to convert into pluripotent cells are transcription factors, which are responsible for reverting it back to a pluripotent stem cells. So majorly there are 3 transcription factors. So these transcription factors are SOX6, OCT4, CMYC, and CLF4. These are the 4 transcription factors which are responsible for the we can revert back pluripotency of multipotent or unipotent stem cell. So what does SOX2 and OCT2 do? They activate NAGON, which is other transcription factor. So this would be the 5th transcription factor, which establishes pluripotency and blocks the differentiation. So SOX1 and OCT4 together activates NAGON, NANOG, which established pluripotency and will block the differentiation stage. CMYC opens chromosome and make it accessible. So it opens the chromosome and make it accessible to the 3 transcription factors, SOX2, OCT2, and NANOG. nanotranscription factors, and CLF4 prevents the cell death. So if CLF4 will prevent the cell death, then those cells can also gain its pluripotency. So this is an idea, and it was discovered by Yamanaka, and thus it is also termed as Yamanaka factors. So this was all about stem cells and pluripotency. In the next lecture, we will discuss about competence induction and competence, as well as Island's induction and Island's induction is in xenophus. We will talk about those topics and other developmental biology basics. So if you like this video, please give the video a thumbs up and do subscribe to our channel for more such content. Thanks for watching.